All right, you guys, 12:50. Let's go ahead and get started. Okay, so a couple of announcements right up front. All right, so the grades are in, and they were a little low, more like the first one. Okay, so the average was about 65, so we're going to give everyone 10 points. Okay, so if you already got your score, that's the raw score, and you can actually add 10 points to that. Okay? All right, so that actually brings everybody up to where they should be. We did have someone get a 98. All right, so that person will actually get some extra credit points.
So just be clear, the 10 points are not
Just to be clear, the 10 points are not added yet. If you did take the exam outside of the classroom, then it hasn't been entered yet, but it will be entered today. Okay? So, all right. Let's continue on, you guys. Exam, just let me know. And we can talk about it after class. Okay. All right, a couple of other announcements, you guys. I am recording this particular lecture. All right, so the final exam. The final exam is still scheduled for next Friday. So your first opportunity you'll be able to take it in class, and then I have actually reserved this room for the hour after the class, so you can take the exam the first hour, and then the second hour you actually would take a makeup exam. If you don't want to take a makeup exam, then you don't have to. You would leave after the first hour. If you plan on taking the exam on the 10th, however, you do not have to come to class on the 4th. Does that make sense? Yes.
Could we come to class on the 4th and do a makeup exam that day and then take the final on the 10th?
So you want to take the makeup exam on the 4th and then come back on. I'd let you do that. That's fine. Yep. Okay. However, you will probably need to come for the makeup exam the hour after because you'll be talking about it in a group. Yeah. And so just to keep it quiet for the final exam.
Is it open note or is it just talking?
Just talking.
Okay.
Just discussing. Yeah. All right. Any questions, you guys? All right. So, yes, question. Yes. Then you will have an opportunity to take the makeup on the 10th. Okay, yeah. If you work on May 4th, you will have another opportunity to take the makeup exam on the 10th. Okay? All right. Okay, so let's go ahead and, this is where we left off. Last time we actually talked about the anatomy of the small intestines, the pancreas, and how the gallbladder, how it's all organized. Here's where we're going to pick it up, okay? Maybe, or you may or may not have known this, but there's a lot of hormonal regulation when it comes to the intestinal phase. In the gastric phase last time we talked about gastrin. Remember, gastrin is released by those G cells. It's easy to remember because G is for gastrin, okay? The G cells are located in the stomach, and when we get into the gastric phase and there's distension, you'll start to see gastrin released from these G cells. You can actually see that here. G cells release gastrin. That increases secretion of acid from the parietal cells, and it also triggers those ECL cells to release histamine. Okay, so that's another big part of this process. However, once you start to get a little bit of food in the intestines, right, the duodenum here, that's when you start to get feedback to slow gastric emptying. You can see that minus sign right here. So you start to get a little bit of food into the small intestines. It feeds back, right? So you start to get an decrease in gastric emptying. Basically what happens is this pyloric junction starts to constrict, and it just starts to piecemeal food into the small intestines. It slows everything down. It slows down muscle motility in the stomach. It decreases acid secretion, and it decreases gastric emptying. So now we've entered into the intestinal phase. Okay, so if you weren't here on Wednesday, I do have a recorded lecture I actually need to put online. It's not online yet. But please review the difference between the cephalic phase, the gastric phase, and now we're talking about the intestinal phase, these three phases. All right, so what happens is a little bit of food actually enters into the area, into this upper part of the duodenum. And now you can see that there's a number of different cells that sense things like acid, lipids, glucose. So that's what we're going to talk about right now. Let's start off with CCK. CCK is a molecule, the name for it is cholecystokinin, CHOLECYSTA, so cholecystokinin, KININ. CCK. I will refer to it on any exam as cholecystokinin with a parentheses that says CCK. Okay, so these cells are actually called eye cells. Okay, so eye cells are also called CCK secreting cells. Eye cells, they detect lipids, so they'll detect any fat actually entering into this area. It senses the fat, and then it triggers the release of CCK. So it's actually a really elegant system, because then CCK enters into the bloodstream. It is a hormone, and it travels to both the pancreas and the gallbladder. Okay, so CCK causes the pancreas to start to release lipases. Those are enzymes that break down fat. And it causes the gallbladder to contract. Later on, we'll talk about what bile does. Just in general, bile helps to break down fats in the intestines. So bile is actually secreted into the intestines, and it's an amphipathic molecule that surrounds lipids. Okay, so this is what it does. Have you guys, bear with me, have you guys ever played around with salad dressing, oil and vinegar, right? So you know when you actually first put the oil in the vinegar, it's like a big globule, right? Big old globule. And then when you shake it up, shake the oil and vinegar up, right? When you shake it up, all of that big fat globules, they go into smaller, tiny little fat droplets, correct? They get kind of small. What bile does is bile surrounds the fat dropules, the smaller fat dropules, so they can't reaggregate. They can't go back together to form these big globules, okay? So they surround the smaller fat droplets and they prevent reaggregation. That allows for the lipase, that enzyme coming from the pancreas, it gives it more surface area. It allows for that enzyme to get in there and really break down fats. Okay, so a very elegant system when you think about it. CCK, right? These CCK secreting cells called eye cells are detecting the fat. They're releasing CCK, which is signaling the pancreas to release lipase and the gallbladder to contract to release bile, all in an effort to break down fats. Pretty cool. All right, so let's go to the next set of cells. These are called secretin secreting cells, or S cells. It's not very creative, but it's true. S cells are cells that secrete secretin. All right. So what secretin does, okay, so these cells, these S cells, they detect acid in the small intestines. They detect the lower pH, the acid that's coming in from the stomach. They secrete secretin. And that secretin is a hormone that travels to the pancreas and signals the pancreas to release bicarbonate. Makes sense, right? Acid is entering into this area. It signals the pancreas through secretin, and then it causes the pancreas to release bicarbonate to neutralize the acid in the duodenum. Cool. Nice feedback mechanisms. All right, and then finally these GIP secreting cells, they're detecting glucose, right? They're detecting glucose or carbohydrates in the small intestines. And just like CCK and secretin, it feeds back both GIP, CCK, and secretin to decrease gastric emptying, decrease acid secretion, okay, and decrease peristalsis, the muscle tension and muscle motility. Okay, so it used to be called gastric inhibitory peptide, GIP. Now get this, it's called gastrointestinal insulinotropic peptide. And so it actually gives you an idea of what's happening. It is detecting glucose dependent, insulinotropic peptide. So it's detecting glucose and then it's signaling the pancreas to start to release insulin. All right. So very nice feedback mechanisms, all hormonal regulation involved with that. Okay, so let's get your time for a while. You guys remember how to do that. See how many of you guys were awake during that small little lecture there. Let's try a real easy one. This first one is acid secreting cells are also called this. So acid secreting cells are in the stomach. Acid secreting cells are also called A, chief cells, B, parietal cells, C, enterochromatin-like cells, or D, goblet cells. A little bit of a review. All right. Give you guys about 10 more seconds. Does anyone need a little more time? Okay, sure. You guys pretty good in the back there? You guys need any more time? One more. Okay, all right. One more, right? Is Mariah, you're good? Okay, so come up to me, come up after class, and I know you're here. So we'll record your answer. All right, so what is the correct answer? The correct answer is B, right? Parietal cells. How'd you guys do? Sweet. 88% of you guys got that right. Most of you guys did. What do the chief cells secrete? Yes, the precursor to pepsin, right? It's pepsinogen. Yep. So pepsinogen is then converted to pepsin by acid. And then pepsin is an enzyme that starts to break down proteins in the stomach. So chief cells secrete pepsinogen. Enterochromatin-like cells, what do they secrete? Enterochromatin-like cells. This is the trifecta that I was talking about. Histamine, yes, good job, histamine. So the enterochromatin-like cells, they secrete histamine. What about goblet cells? What are they? Oh, we didn't talk about goblet cells yet. Mucus, mucins, yeah. Okay. All right, how about this one? This one's easy. G cells release the hormone. I'll let you guys answer. This will be a speed round. Speed round. All right, 10 more seconds. See if we can get more than 91. That's what we got before, I think. Sweet. All right, good job. All right, so I'm going to go ahead and pause these submissions. And the correct answer is, of course, A, gastrin. Yes. 94% of you guys got that right. Nice. All right. Eye cells. This one's a little more tricky. It's not as intuitive. I doesn't stand for anything. Speed round. All right, doing great. 10 more seconds. Can we get one more? I think we got 94 before. All right, I'm going to go ahead and pause this one. And the correct answer is C. Nice, you guys are doing great. Okay, oops. Let's see, I think we have one more. S cells, you guys know, are secretin, so I'm going to skip that one. Oh, sorry. What about this one? CCK has effects on. I talked about two of these, but there's one I didn't talk about. Oh, it's still not? Oh, what? Oh, okay. So the answer to the one before was S cells secrete secretin. That's pretty easy. S for secretin. Yeah, you guys can talk in your group. Don't. All right, we're up to 87. 10 more seconds. Great. Okay, we got more than any of the other ones here. Perfect. Okay, I'm going to go ahead and pause. Everybody had a chance to answer this one? All right. What? Somebody said no? Oh, perfect. Okay, so the correct answer, hopefully you guys got that, was D. All right. Okay, so CCK, cholecystokinin, has effects on the gallbladder. It causes the gallbladder to contract. It signals the pancreas to release lipases, and it relaxes the sphincter of Oddi. Remember, the sphincter of Oddi is the opening. Those are the vessels from the gallbladder and from the pancreas. It has a common sphincter called the sphincter of Oddi. Okay? All right, good job, you guys. So let's go back here. Let me just show you. I do want you guys to know this diagram. This is one of the ones that you'll probably need to label. Here's the sphincter of Oddi right here. It relaxes the sphincter of Oddi. All right, doing great. Okay, okay. So, so here are the different hormones that we just talked about. This is a very nice table that summarizes all of it. We're mainly talking about what's happening in the stomach and the pancreas and the gallbladder. Okay, so also CCK relaxes the sphincter of Oddi. So if you want something to read, this is just a nice summary slide, a table that summarizes pretty much everything that we've talked about. You don't need to memorize this. This is just for future reference. The one that we were talking about, I told you, was gastric inhibitory peptide. It's now called glucose-dependent insulinotropic peptide. So there is a name change with this one too. Again, just remember to refer to it as GIP. This is the older name, and now they have a new long name associated with it. Okay, so we also talked about somatostatin last time. Remember, that's the only hormone that inhibits histamine release and acid secretion from the parietal cells. So later on, we'll get to peptide YY when we talk about appetite. So again, a nice summary slide of a lot of different hormones that control digestion. These are also just for your reference when you actually go home. It actually gives you some information about these different molecules like fat, amino acids, acid in the duodenum, and how that increases the secretion of hormones that enter into the blood. This is plasma enterogastrones, and then they all have an effect on decreasing gastric emptying. Helps to slow the gastric motility and secretion, which then allows for adequate time for digestion and absorption. Okay, so these are redundant. Some people like to see this in this form. This just talks about secretin. This one is about CCK. So again, if you just want to go through these flow charts, it just gives you more, actually gives you the same information that we already talked about. Okay, so let's go ahead and move on. Talk about the anatomy of the small intestines. Then we're going to go into the motility of the small intestines and then function again. We're going to go into more detail on the function. So this is a nice slide kind of giving you some detail of the anatomy of the small intestines. Okay, so you have the mucosal layer. That is the layer closest to the lumen of the inside of the tube, the GI tract. The inside, so this would be the inside of the small intestines. And the serosal side is the closest to the blood supply. Okay, so there's 4 different layers: mucosa, submucosa, muscularis layer, and then the serosal side, again closest to the blood. The epithelium is actually organized in these villi. We'll talk about villi and microvilli in just a second, which increases surface area. So the more folds or undulations, the more that increases the surface area to maximize absorption. So then you have some connective tissue, lamina propria, and then muscle layers right underneath that. You have a muscle layer in the mucosa. The submucosa is where all the blood and lymphatic vessels are, and it has its own nerve plexus. And then right underneath that is the muscle layer that has circular muscle and longitudinal muscle, and then a different nerve layer, myenteric nerve plexus. So, again, I'm just going to reiterate this. Your intestines are the most innervated organ in your entire body. They have more nerves than any other organ. And that's because motility is a big part of its function. Now, if you actually have acid, that maybe you have too much acid that's being produced by the stomach and it starts to eat away the epithelial cells, usually there's a nice mucus layer that traps bicarbonate. So if some of the acid gets by that mucus layer, and it'll meet bicarbonate and be neutralized. But if there's so much acid, it starts to eat away the epithelial cells. That's an ulcer. You guys have probably heard of a duodenal ulcer before. This is what that looks like. In this case, the ulceration has degraded the epithelial cells all the way to the submucosa, where you see visible blood vessels. Now, the way to diagnose this is they actually have to put a camera, an endoscope, down your throat, past your stomach, and then take some pictures of your small intestines. That can reveal ulcers or complications. Okay, so zooming in on the anatomy, we're still sticking with the anatomy of the small intestines. You can actually see that it's organized in these large folds. This whole thing right here is called a villus. Multiple are villi. And then within each villus are all of these smaller microvilli, right? You can count them all up. These are all microvilli, also undulations. So these are known as brush borders, and they fringe the villi to further increase surface area. Again, this is all about maximizing absorption. Now, so you can actually see how each of the villi are organized. You can see the blood vessels. They actually go into the villus, the arteries, arterioles, the venules, and this green tube right here is the lymph system. And then you have the capillaries up here surrounding the lymph system. Now, when we talk about fat absorption, fat, those chylomicrons, they actually package it in a certain way and they deliver it right into the lymph system. A lot of that fat goes into your lymph system. Okay, so I'm going to go into a little bit more detail on the cell types that are lining the small intestines. That's the next slide. This gives you can see these are the villi. Each one of these is a villus. But it's giving you more detail about each of the different cell types within the villus. So you already know something about, this is those undulations, folds are increasing surface area. There's the four main layers. And here are the mucosa cells. Enterocytes are absorptive cells. They absorb stuff. Carbohydrates, proteins, amino acids, and lipids. This was one of our top hat questions. Goblet cells secrete mucus, mucins. And those mucins actually help to protect the epithelial cells. There's also bicarbonate secretion that, again, if any acid is actually around, it gets trapped in the mucus and then neutralized by the bicarbonate that's within that mucus layer. Enteroendocrine. Endocrine is all about hormones. So these enteroendocrine cells secrete hormones. And those hormones are like CCK, secretin, GIP. And then Paneth cells, these are a special type of cells that secrete antimicrobials, lysozymes, things like that. Okay, so you'll also notice this crypt. The small intestines are organized with these deep valleys right here. These are called the crypts. And crypts are where the stem cells are. These cells are stem cell-like. So they replenish the cells of the villi. So they start to, like new cells are formed down here in the crypts, and then they move up towards the top of the villus. So you don't need to know this for the exam, but there's different viruses. Like you guys may have heard of rotavirus, or maybe not. Some dogs can get rotavirus. Farm animals get rotavirus. Rotavirus, causes diarrhea. It causes intestinal disturbances, and basically, and TGE is another one of pigs. You guys maybe know pig TGE, transmissible gastroenteritis. Those two viruses, they attack cells at the top of the villus. Okay, so not so deadly, not so bad. I mean, they can recover from that after a few days. However, there is another disease of dogs. It's called parvo. Anybody heard of parvovirus? Deadly, right? It is so potent. Parvovirus, you only need 100 particles for that dog to die, for it to be a lethal dose. And it causes bloody diarrhea. The dogs can literally, it usually attacks puppies, and they usually die within days after contracting parvovirus. It's so deadly because it actually attacks the crypts, right? So what happens is it gets into the crypts, and then they don't have any way to replenish the epithelial cells of the villus. And it's very lethal. It's very deadly for dogs. So that'll help you with understanding what crypts are. Okay, again, surface area is really important. Nutrients are hydrolyzed in the lumen of the GI tract. The uptake of end products is very slow, so it has to be fast enough to move the bulk bolus, the food, but slow enough to actually help with the maximize absorption. Okay? So there's two ways to improve surface area. The gut undulations, those are the folds. I started with the bottom one first. Those are the villi. These are the folds. And increasing gut length. So the small intestines is very long for a reason, to help maximize surface area and absorption. Again, just as I said before, it has to be fast enough to minimize amount of indigestible material, but slow enough to allow enough time for digestion and assimilation. And it's all controlled by nerves and hormones and smooth muscle. Okay, so moving right into the 3 types of motility in the gut. I do need you to know these. They're called segmentation, and you guys already know peristalsis. Segmentation, peristalsis, and then what's called MMCs. I'll come back to this. MMCs look something like this. They're migrating myoelectric complex. MMCs. Migrating myoelectric complex. And I'll come back and talk about this. But those are the 3 types of motility in the small intestines. Okay. What is segmentation? Segmentation is when there's alternating constriction and relaxation of the tube, of the small intestines. So here's segmentation. You can see at this particular point right here, you get constriction, and right next to it, dilation. And then the next minute, you get dilation here and constriction. So if you can actually see my hand movements, segmentation is about mixing, right? Relaxation, constriction, and then it's an alternating relaxation, constriction, right? All the way down the line. It's called segmentation, okay? And what you're doing is you're mixing and breaking up those food particles, mechanically mixing and digesting. Peristalsis is about moving that bolus of food down the line. Remember the circular smooth muscle. constricts behind the bolus of food, my sweatpants analogy, and then the longitudinal muscle shortens the tube itself. So it pushes that bolus of food down the line, peristalsis. It's a coordination, again, between circular smooth muscle and longitudinal muscle. So, again, this is just another slide showing you segmentation. You can see these two boli of food. And as segmentation occurs, it breaks them up, and then it starts to mix them. Okay, so peristalsis then helps to move the food down the line, downstream. Now, with this type of motility, obviously nerves are really important. You have that myenteric plexus and all of those neurons. This is in the muscularis layer. You can see the circular smooth muscle and the longitudinal muscle. And peristalsis is actually controlled by very specialized cells called the interstitial cells of Cajal. He's a Spanish scientist, Cajal. And what's really interesting about this is that the interstitial cells of Cajal have pacemaker cells, just like the heart. They act as pacemakers. And I'll show you this in the next slide. You can actually see there's an eroding depolarization until it reaches threshold, and then you have these spike-like action potentials. These are calcium spikes. And then it relaxes. Again, because of those funny channels, you get a slow depolarization. And the longer it's above threshold, the more calcium spikes, which causes constriction of those muscle cells. Remember, an increase in calcium causes constriction of smooth muscle cells. So this is kind of bringing home a lot of things that we've talked about. Action potentials, threshold potential, and then smooth muscle cells. After the intracellular levels of calcium rise, you get these nice contraction, right? And the more calcium spikes, the greater the tension, the greater the force. All right, so going back to the previous slide, resting muscle tone controls lumen diameter, and it's regulated by intrinsic pathways. It's a myogenic pathway and neurotransmitter release. Okay, so what is migrating myoelectric complex? This is interesting. I think we may all kind of think about this after we eat a meal. When you're resting, this actually goes for 90 minutes. It's a very long process. But I would say a couple of hours after you eat a meal, you have these migrating myoelectric complexes that actually sweep through the entire small intestines over a 90-minute period to sweep any remaining indigestible food into the colon. So it occurs a couple of hours after we eat, and it starts in the stomach. You can see the motor activity starting in the stomach and then sweeping through the duodenum, the jejunum, and then the ileum, pushing that last remaining indigestible food into the colon. All right, so let's go back and now talk about some of the function of the small intestines. We have to get into a little bit more detail on how some of the lipids, carbohydrates, and proteins are broken down. Okay, so remember that anatomy is really important. I've kind of already talked to you about memorizing this particular figure. Going back up to the duodenum. Now, I also want you to realize that in the pancreas, you have this pancreatic duct. Let's blow this up. And these are typical exocrine glands. They secrete hormones, which is delivered into a duct into the pancreatic duct here. Now behind that, these are the endocrine cells of the pancreas, like the beta islet cells that secrete insulin. So this is a typical acinar gland. Remember the acinar glands? They have this bulb-like projection, just like salivary glands, right? All right, so it's the duct cells that actually secrete bicarbonate, and that's through those CFTR channels. CFTR not only transports chloride, but also bicarbonate. So usually people who have cystic fibrosis, they are diagnosed with cystic fibrosis because they have plugs, they have a mucus plug of the duct cells of the pancreas. So even the mildest form of cystic fibrosis, they actually have to take pancreatic enzymes as supplements because they don't have the ability to secrete pancreatic enzymes into the duodenum. And that's because they are missing that CFTR channel, which transports bicarbonate, causing it to plug up, actually. Bicarbonate is transported, and then water, it keeps it open. Otherwise, without that channel, you get these mucus plugs. All right, so some of the enzymes that are really important. First one is trypsin. Trypsin is a molecule, an enzyme that breaks down proteins. And the pancreas actually secretes it as a precursor called trypsinogen first. Otherwise, if it actually synthesized it in its active form, it would digest the duct and the epithelial cells that made them. So you have to secrete it in an inactive form first. It gets delivered into the duodenum, and then there are membrane-bound kinases, enterokinases, that convert it from trypsinogen to trypsin. And then trypsin is the active form. Trypsin then starts to break down proteins in the small intestines. Anybody remember the enzyme that starts to break down proteins in the stomach? We just talked about it. Pepsin, right. So chief cells secrete pepsinogen. That gets converted to pepsin by the acid that's in the stomach. Then you have a second enzyme, trypsin, that further breaks down proteins in the small intestines. Trypsin and chymotrypsin are different types of enzymes that break down proteins. Carboxypeptidase is also another one. All right, so keep going with these enzymes, keeping in line with this line of thinking here. Lipases, as we talked about, are released from the pancreas triggered by CCK, releases fatty acids from triglycerides and phospholipids. Proteases like trypsin and chymotrypsin, carboxypeptidases break down proteins, and amylase breaks down polysaccharides into oligosaccharides, few. Oligo means few. Okay, where else is amylase secreted? Anybody remember? The saliva, yes. So you have salivary amylase and you have pancreatic amylase, further breaking down carbohydrates like polysaccharides. And then nucleases break down DNA into nucleotides. So you've got mechanical digestion, which we talked about with motility and segmentation, and you have chemical digestion. Okay, so also I want to make sure that you guys understand a little bit about the microbiome within the stomach. This is a really hot area. There's many animals that benefit from the assistance of symbiotic organisms, bacteria, fungi, algae. But the three main types that participate in digestion, enterosymbionts. Enterosymbionts. They live inside the small intestines. They live within the inside of the tube. I would say instead of exo, ecto. That's probably a better term now. Ectosymbionts actually are actively cultivated outside the body or in cavities, like the mouth. Whereas endosymbionts are embedded in the host cells. Good examples of endosymbionts are mitochondria, chloroplasts. They once were individual organisms, bacteria. All right, so now let's talk a little bit about how food is absorbed, right? How is it brought from the inside of the small intestines into the blood? How is it absorbed into your system? There's carriers. And as we know with the carriers, polar molecules require proteins. This is more facilitated diffusion. If they're going down their concentration gradient, again, facilitated diffusion, but against their concentration gradient, you need a secondary active transporter or ATP and a primary active transporter. Also, sometimes vesicles are formed if you're engulfing certain materials. Pinocytosis is the same thing as cell drinking. Cell drinking brings in insoluble nutrients. And cell eating, technically, phagocytosis, is when you have larger particles. And then the reverse of that, not engulfing but expelling, is exocytosis. So if you've taken a general biology course, you probably know some of these terms, but this is really how nutrients are transported across plasma membranes. All right. So this is something that you can put on your slides. You're not going to have to regurgitate this in an exam. This is just for your own kind of information. So this is giving you an idea of how starches, cellulose, actually we don't have the enzymes for cellulose, but disaccharides, glycogen is broken down. It starts to break down in the mouth, and then the pancreatic amylase breaks it down from disaccharides to monosaccharides. This is giving you more information about what sucrose is, glucose and fructose, lactose, but this isn't something that you'll need to know for the exam. All right. I think we will leave it here, you guys. We'll finish up on Friday. We'll do the active learning exercise on Friday. And this will only take another 15 minutes or so to finish up lecture 16. So I will see you on Friday, you guys. I'll see most of you guys in lab tomorrow for your presentations, and I'll stick around for any questions that you have.
I don't know if you remember we spoke about that. I went to the website, but it actually says like it's a download.
Oh, I know. I saw that. He wrote me an email yesterday. So I'll send you the flyer when I have.
Okay.
Okay?
Thank you.
You're welcome.
So I missed everything about the final exam and everything.
Yes. Okay, so we still have an opportunity to take the final exam on Friday of next week. If you plan on taking it on May 10th, you don't need to be here on May 4th.
And if I take one on next Friday, it would just be this time.
Yes, and you don't have to come on May 10th.
Oh, it's like I probably. And what does the final cover? Is it just the GI?
Yes, GI and endocrinology.
Okay,
Yep.
and then like that's what the reading is. Okay,
Yeah.
so it's all right. That means it.
You might be
So
done
I
by
just
next
essentially
Friday.
I essentially just need to study for that and whatever one I want to retake.
Exactly.
Okay,
Yeah.
thank you.
Awesome.
Will there be a review next Friday?
Yes, it probably will be. Okay, so you guys have your practical on Thursday.
Yeah.
So I'm thinking it probably the same Wednesday after class. Okay.
Perfect.
Okay.
All right. I missed the exam, so I was
Okay.
on for that funeral.
Yes.
I could do it Friday morning if that works for you.
That does.
Okay, perfect.
Okay.
Like 10:30?
What time? Yeah.
Is that good?
I'll put you on my calendar.
Thank
Okay,
you.
Yeah.
Hi. So I did turn these in because there was like a snow day. I
Oh,
emailed
yeah.
you about it.
Yep,
Do
yep.
you just want these now then?
Yeah. Why don't you hand them to Julie?
Okay, thank you.
Yeah. Thank you.
So I had emailed you a couple of times
Okay,
about like the question number for the internship course.
Yes, and I will get that to you.
Okay.
Okay.
Okay. Just
Yes.
reminding
I
you.
haven't forgotten.
Okay.
Yes.
Thank you.
I will do that.
We had a couple questions about the video.
Oh, yeah.
We're kind of confused where to start.
Oh, okay.
About the
We were
entire
like
thing.
thinking, finger
Okay.
puppets or something. Okay, so you guys had a script all set out.
Yeah.
Okay, so I would go ahead and start building the slides and just thinking about the script. Like, it's okay to read from a script. as
Does
you're
it
going
have to
through.
be like taping us? Like
No.
we don't have to be.
So it
It
can,
can
be
be
like
a
a
power-
Camtasia type.
Yes.
That can be a PowerPoint too.
Or PowerPoint and
cool.
voiceover.
Oh no way!
We just weren't sure because the example you showed was very interactive, and we were
Yeah,
like,
you can do that, or
Okay.
you can just do a simple PowerPoint slide.
Cool.
Okay, so I just really want to make sure that, so this is your audience. Next semester, the students that are going to be in this class, these will be short little tutorials to help them with material I
Right.
probably won't cover in class.
Oh.
So yeah, so if it's anatomy related, or just think about it in terms of you're explaining to the next semester's class some of these
Cool.
things. Yeah. 5 minutes, just an educational tutorial. The best ones I'm going to highlight on next semester's,
Totally.
Canvas
Alright.
site. And
Perfect.
yeah.
Perfect. Thank
So
you.
make it
That's
fun
very
or
helpful.
funny or,
Cool.
whatever. Yes.
Thank you very
Yeah,
much.
you're welcome.
For one of the into thin air, I have a 0 on Moodle, so I'm assuming I remember you said that people didn't put names on them.
Yes.
Um,
Okay, so I have some.
yes.
I have to bring them back.
Okay.
I don't think I brought them today. Did I? Maybe I did. No, I did not. So I will bring them back. I have them.
Should I just email you?
Yeah.
Okay.
Or Julia, can you remind me to bring back those no names
Okay.
on Friday? Okay. I will remember. I'll
Okay.
just put them in my bag as soon as I find. There's 2 piles. I'm
Okay.
sure they're in there.
Okay.
Okay.
thanks.
You're welcome. I got your email,
Oh,
so
good.
sorry I didn't have time to.
Oh, that's okay.
So it was for 4096, and you
Yes.
wanted to know whether you needed to
Oh,
about the presentation.
I just couldn't submit it on the Moodle
Oh,
site.
that's
It was
right.
too large,
Yes,
so
yeah,
I
yeah.
just emailed it to you.
Yeah,
Is that okay?
or even if you have it in Google Docs, you can just share it with me.
It's not. It's
Okay,
just in
no worries.
PowerPoint.
Okay,
You should
yeah.
be able to see it. If you can't,
I'll let you know.
then yeah. And
Okay.
then I don't have a stapler, but this
Okay.
is my journal.
Oh yeah, awesome.
So do you want me to just fold the corner?
Yeah.
Okay.
Yeah. No, that's
Thank
perfect.
you. So
yeah.
I should have everything submitted.
Yeah. Thank you, Lily.
Yes. Thank you.
Awesome. Thank you so much.
I won't be here on Friday.
Oh, okay.
What's the active learning
Oh.
thing you were saying?
Yes. Okay. So that. Okay. Yeah. No worries. Okay. So that is.